Aditi.
Yes, sir. So a very warm good afternoon to one and all present here. I, Aditi Sharma, on the behalf of Dr. Vital Rao V.K. Patil's Medical College and Hospital, would like to welcome you to this International Research Conference 2022. I now would like to call upon the organizing secretary of our research cell, Dr. Sandeep Kadu, sir, to say a few words and begin this session. So,
Good afternoon, all of you. On the behalf of Dr. Vital Rao V.K. Patil Foundation's Medical College and Research Cell, I, Dr. Sandeep Kadu, Research Cell Coordinator, welcome all of you. This International Research Conference is a brainchild of our respected Dean, Dr. Sunil Nathamaske, sir. Sir said that we have to organize an international research conference. 2 years back, in 5th March 2020, we have organized National Research Conference. It was a grand success. After Sir's suggestion to organize International Research Conference, I took help my beloved friend, Dr. Suhas Khanwe. He is the President of Clinical Services at Acutes Research Laboratories, Navi Mumbai. With his help, Dr. Michelle Ma'am has consented for delivering a session on the global perspective of clinical trial. I'm very thankful, Dr. Michelle Ma'am, for her valuable time. Also, my friend Dr. Anand Gaikwad from Australia also consented to deliver a session. And my 11th standard Ferguson College friend, Dr. Akash Pandre, is now Associate Director in a Johnson's at Fort Washington, USA. He also consented to deliver the session on psychopharmacology of bupropion, a novel discovery and potential implications. Actually, it's our comfort time. That is, now it's the 3:30 p.m. in India, but all are from different countries and their timing is different. Still, they have consented to deliver the session. I'm again very thankful from our institute for their valuable time. Before wasting any time, I request our respected Dean Sir to enlighten us with your knowledgeable thought. Sir, please.
Warm welcome to all the dignitaries and participants. Firstly, on behalf of Dr. Vital Rao V.K. Patil Foundation's Medical College and Hospital, Ahmednagar, I welcome all in this research conference, which we are organized on the international level. For the sake of knowledge of the dignitaries, our college is MBBS having 200 seats, which was started in 2004, along with postgraduate courses in the 10 disciplines. Also in our institute, we have PhD courses run in 3-4 departments. Also, some of our faculty are already completed the PhD and some of our research scholar. In our institute, we have established research cell for the promotion of the research in the institute. Also, we have established a very good research museum also. 2 years before, we have conducted National Research Conference also. And this is the attempt to conduct the International Research Conference with the present dignitaries. Everybody knows that tomorrow's word is of the researchers only. And keeping in this line in mind, we are motivating our graduates for the angle of research. So I will not take much more time. I wish all the best for all the dignitaries, those who are giving the best lectures on the research. All the participants will be 100% benefited with this knowledge of the research and they will be motivated positively. I am very thankful for Research Cell for organizing such a wonderful CME and getting such a good eminent speakers. Thanks once again.
Aditi, please.
thank you, sir. Thank you, sir, for sharing your knowledge with us. So, good afternoon, everyone. Now, I would like to introduce you all to one of the ideal examples of women in power, Michelle Yanji Jiang. Michelle is the Chief Development Officer of the Trident Pharmaceuticals, which is an African-based unit of the Fosun Pharmaceuticals. Michelle completed her bachelor's in clinical medicine from the Shanghai Medical College and her MBA from the Washington University in St. Louis. She works in collaboration with the pharmaceuticals for the medical marketing to develop anti-malarial strategies, products, and a lot more stuff. She understands the dynamics and the diversity of the world and has conducted several bioequivalent studies, clinical studies for strategic planning of the anti-malarial programs. She is adamant to bring innovative and affordable medicine and healthcare products to developing countries to improve life care. We are very, very happy to have you on board, Michelle ma'am, and wholeheartedly welcome you to this international research conference.
Thank you. Thanks a lot to the organizer for inviting me. So I will start my my talk. I'll share the screen. You can see my screen, right?
Yes, ma'am, we can.
Okay. Sorry, just a moment. Okay, so my topic today is about the global perspective for clinical trials, but which is a very big topic for me. So I will give my talk according to my experience and some views from the industry perspective. So the development of a new drug and to launch the product in the market is just like to give the birth of a baby. You have to be patient and to wait for a long time for the time and to experience different stages. And like for the baby after 9 months, you have the baby, and after maybe a long time for the drug development, you will have the product ready to launch in the market. But we also have to know that just like not every baby can be alive after 9 months, so not every product can be successful in the market. So, so in the drug development period, you have to experience 3 stages, which is the first stage is the drug discovery or the discovery of a molecule, and then you have to experience the preclinical stage, and then if the drug still works, and then you go to we move to the clinical stage, and then this drug development process is a very long process, which is it will normally take about 12 years. There's no doubt required to pass through this painful and stressful time, money, to ensure that the quality product can be launched in the market and can be used by the patient. Because after all, the product is going to get administered to the human being, and any wrong or faulty product can result in life-threatening hazards. So we have to be very careful. And then the drug development, again, so it is a very long process. You will see here. So basically, on average, from the beginning of you have a molecule to the end when for the product to launch. Normally, it will take about 12 to 15 years, and 50% of this period, about 6 years, will be the molecule development and the preclinical stage. So I give example here. At the beginning, we have 5,000 to 5,000 or maybe 10,000 candidate molecules at the beginning, and then go to the preclinical stage. We can only have maybe 250, and it is 6 years after. And then with this 250 lucky candidates, we move to the clinical stage. And then we will have to experience 6 to 7 years to go through the phase 1 and phase 2 and the phase 3. So in phase 1, from the 5,000, we only have 5 lucky candidates, and then maybe at the phase 1 stage. But when you finish the phase 3 studies at the year 6 or year 7, and then probably you only have 1 product. And this product, with all the documents and data, the company prepared the drug for the submission to the drug authority for marketing authorization approval. And this evaluation period will again cost about 2 years. And then if the drug is lucky enough, pass through all the studies and the authority evaluation, and then at the last stage, after 12 to 15 years, we got 1 drug ready to market. But so this is a very long and painful stage and can be failed at any time in this period. And then so we have this question, why the success ratio is so low? So we have 5,000 candidates, and probably, if lucky enough, we can have 1 drug launched in the market. So we see here, so around 10% molecules failed in the animal toxicity preclinical stage, and then about 40% fail again in the pharmacokinetics stage, which is preclinical, and then go to the clinical stage, and then you have 10% possibility to fail because of the adverse effects in human subjects. And then 30% of the molecules will fail again in the lack of efficacy. And then if the drug goes through all the steps and still it's possible, it could be the candidate could be a very good drug, but because of this, something happened at the last commercialization stage, and then maybe like the company is bankrupt or for some other reasons, and then still the drug cannot be launched in the market. So 5%, and then you have another 5% unexpected situations. So anything can be happen in this period in the drug development. So with very high risk of failure. And then this picture, this slide explains again the process of for drug development. So at the beginning, you have this at the basic research stage, we have 5,000 candidate molecules, and then when the molecules go to the we have 250 can go to the preclinical stage, and then maybe we are lucky enough we have 5 candidates can go to the clinical trial stage, and then after 10 years or so, and then we have 1 molecule, and we go to the drug authority for marketing authorization application. And then after 2 years, and then we have the drug launched in the market. So this is the regular process, the normal process to develop a new product. But especially I think during this 2 years COVID time, we realize that there also has a kind of accelerated drug approval process. The difference is again at the clinical trial stage, because for the regular process, a molecule have to go through the phase 1 and phase 2 and phase 3 trial to collect enough data. But for some emergent situations like the COVID, and then there's a huge demand for new treatment, and then this phase 2 and phase 3 trial can be skipped or combined together. So you have, so which means after this a simplified process, and then a clinical trial process, and then the drug can go to the drug authority for marketing authorization application, or this conditional approval process. And normally, if it is like a global pandemic crisis like the COVID, and then the drug authority will do fast evaluation too. So maybe in 6 months, you can have the drug launched in the market. But it is not the end. You still have, the company still have to do different clinical trials to submit supplementary documents, dossier status to the drug authority to prove that the drug is safe and the drug safety and efficacy. And so if the company can do that, which means later probably the drug still has a risk to be, the company will have to be requested to withdraw the drug from the market. And even a new drug is launched in the market, it is not the end of the story again. So the company have to do phase 4 post-marketing trials to study the long-term safety and efficacy of the drug in a huge population. So again, here's this table is about explain a little bit more about the 4 stages of clinical trials and the purpose of these 4 trials. So the phase 1 trial only need a small amount of subjects, 15 to 30, and the purpose is to find the safe dose to prove that the drug is safe for human subjects. And then go to the phase 2 stage, which normally requires less than 100 subjects. And this stage of study is to prove the drug efficacy. And then if for phase 3 trial, it always requires thousands of hundreds to thousands of subjects to compare the new drug, new treatment against the standard treatment to prove that the drug is better or at least as good as the already in market products. And then the phase 4 trial, which is the post-marketing clinical stage, and it will be done on several thousands of patients, and just to prove the long-term safety and efficacy of the study, because the normal, phase 1 to phase 3 trial, the period is relatively short, and the population sometimes is not large enough to find out the potential risks. But that's why the phase 4 trial is required. And then, so the drug development process for a company, for the industry, is very costly for money and for time. So again, to develop a drug, to get a drug launched in the market will take about 12 to 15 years. And for a company, probably have to invest around $2 billion just to make a successful drug. Because we also have to consider that even you have 5,000 candidates, probably at the end, you can only have one drug. So the cost of this successful drug will also, include some of the other molecules failed on the way. So that's why the cost is very, very high. And when we evaluate and when we talk about the cost to develop a new drug, you can not only just mention those successful products, but also have to talk about those drugs and the molecules the candidates failed on the way. So that's why the cost is very high. So what is a clinical trial? The definition of a clinical trial. So I will talk a little bit here, and I believe the other speakers later, they will talk more because they know more, much better in this area than me. So a clinical trial from the industry view, we think it is a key step during the drug development, and it is done once the preclinical phase is over and the results are promising. And then a clinical trial is always deals with human subjects, and because it is deal with with human beings, so the safety and well-being of the subjects are always the priority. And then the drug efficacy takes a back seat, because this is the main and the key evaluation points to define a successful drug, a good drug. So the objective of clinical trials is to get the treatment. So there is ways to classify the type of clinical trials. So there's just one example here. So this clinical trial applies all of the products going to play approval. So some clinical trials are for a kind of treatment. So it is to get treatment for disease. So like the antibiotics in infection, and the antivirus drug, the anti-COVID drugs are good example. And then the second type is to get a remedy to prevent disease. The classic example is the vaccine products. And the third type is to get an agent for early detection and screening. So the example is the dye for imaging. So you have, but you can get other ways of classification, such as on the basis of drug product indications, or the different ways of study design. So the key players in a clinical trial, here are the basic elements I have in my mind. So the most important, you need to have human volunteers. They can be healthy subjects, can also be patients, based on the type of clinical trials you are going to do. And then you need to have the medical staff, such as the investigators, the physicians, and then you need the research nurses, and then the pharmacists to manage the investigational product. And then you need the lab technicians and the CRAs, which is for the industry especially. And then we also need the statisticians and or the data managers to manage the study results and to prepare the data, the report. So these people are all important for a good clinical trial or for any kind of clinical trial. And then how a trial is started, I think the other speakers, they will explain more. I just give a brief idea. So when you have the idea, yes, we want to do a clinical trial, and then you discuss this with your colleagues and your peers, and then you have to generate this idea to a concept of protocol. And at the same time, you also have to think about the budget for this trial. And then once you have a draft of the protocol and the budget, and then you can you will organize a meeting, internal meeting, to discuss the protocol and the budget with your colleagues. And then you have to finalize the protocol and the budget, and then submit this package of protocol as well as the budget to the sponsor or to a grant to a grant for grant application. And then once this protocol is approved, and then you will prepare all study related documents, and then submit the protocol to EC, ethical committee for review and comments. And then you also need community engagement, such as the DSMB. And then once you get everything done, everything ready, and then you will start you can start the study. And then this is the same idea of this study preparation process, so I will not go into the detail. So I like to emphasize here that the production is strictly controlled and is to ensure that the delivery of quality study results you will have it at the end. The clinical trial is not about generation of data, but it is about a highly regulated process, and it should be conducted in accordance with the study protocol and with the ethics committee approval and the different guidelines, such as international guidelines, the ICH, as well as the national guidelines, which could be different based according to the country. And but the most important concept, the ICH GCP guidelines, is universal, and it is the ethical and scientific quality standard for designing, conducting, recording, and reporting trial results. And so in the next two slides, we will talk a little bit about ICH GCP, but I think the other speakers, they can explain more. So what is GCP? GCP, is the short name for good clinical practice. So it is an international and scientific quality standard for the design, construction, performance, monitoring, auditing, recording, analysis, and reporting of clinical trials. Provides assurance that the rights, safety, well-being, and confidentiality of human research subjects are secured, and the data and reported results are credible and accurate. So GCP is very important, especially for the industry, because the clinical trials conducted by the industry will normally be used or be submitted to the drug authority for evaluation. So GCP is the key role. And then, so this is just for, I just show you here the list of ICH countries. So you have the member countries and also have the observers, and among the member countries, they have different classes of ICH countries. And just to mention that for US FDA, Europe, and EMA, the European Drug Authority, they have the highest priority in this ICH system. So some few global facts and figures about the clinical trials. This is so today more than we have more than 7,000 medicines are under development around the world, targeting areas of high unmet need. So from this graph, we can see that the leading disease area would be category cancer. So cancer is the leading part, and the second one is neurological disorders. So these two are the most popular targets And then, but we also see that there are a lot of studies focusing on the infectious disease. So I think, especially in the last 2 years, a lot of trials were focusing on the treatment of COVID-19. So this is the trend, and this trend also gives you that what would be the future market potential for different drugs. So again, oncology, and then neurological infectious disease. And so these are the trends. And then this graph is quite interesting because it shows that the total number of clinical trials registered from since 2000 to February 2022 this month. So there is a very interesting point at 2006. So before 2006, the number of clinical trials conducted globally, the number is quite small. But after 2006, you can see there's a very sharp growth after 2006. And so in the clinical trial industry, and we also believe that the same trend is expected for the coming years. So a lot of trials, and this industry grows very fast. And now here is the percentage of recruiting studies by location at very fresh data, February 2022. So we see 34% of the clinical trials are done in US. And then we have 60% of trials done outside the US. And then there's 5% overlap, which are the trials done inside US and outside US. So these are the global trials or multiple site trials. So so from 2020 to 2028, these 8 years, it is estimated that around $42 billion to $60 billion will be invested in this global clinical trial industry with a CAGR of 4.8. So which is a very high rate and a huge market potential. In this picture, this graph is quite interesting because it is this overall consideration for conducting clinical trials in selected countries. So here we see that we know that to do a clinical trial in US, the cost is 2 times of when you do a clinical trial in India or in China. But I'm a bit surprised to see that China is even cheaper compared to India, because this is not what I experienced here, because I'm based in China, but I know that to do a clinical trial in China is much more expensive in India, according to my experience. But anyway, we know that for both China and India could be a good place to do clinical trials compared with US or with Europe, which is much cheaper. So which means for both India and China, there would be a big potential to have this kind of clinical trial industry or third-party clinical trial service in these 2 countries. And then the last part is to talk about the challenges encountered in clinical trials. So from this picture, we see that 85% of the trials fail to retain enough patients. And then 80% of the trials fail to finish on time. And then 50% of the trial failed of site enroll. Probably they have problem to enroll the subjects or even no subjects. And then 40% of the total US pharmaceutical clinical trial budget goes toward recruitment. So the patient still is the key and the trigger part. And then 30% of patients drop out of a clinical trial. So again, from this graph, you will see that the fail of a clinical trial always linked to the patient, which is the most unpredictable part. So to conclude, although the challenges are more and the risks are high, our organized, committed, and sustained efforts will lead to a successful drug development in future. And I'm very confident for the industry and for especially for this clinical trial service industry, which would be a brilliant future. Thank you.
Aditi.
Thank you, ma'am. It was a privilege having you, and I'm sure all of the participants have appreciated the knowledge you have given us. It was valuable information. Thank you, ma'am. Now moving ahead with the next part of the session, I would like to call upon the next speaker for the session. I am very grateful, and it is an honor for us to host Dr. Suhas Khandwe, sir. Sir is the President of Clinical Services Private Limited Research Laboratories from Navi Mumbai, and Sir has completed his MBBS and MD from Pune University. He is a working clinical pharmacologist, and Sir is a pharmacokinetist. He has more than 22 years of experience in pharma industry. Sir has managed different clinical activities for the same, and he has played a role in all regulatory inspections and accreditations in this field. Sir has regulated various biostatics department, and thank you, sir, for joining us today, and it is a privilege to have you. Please go ahead.
Hello. Am I audible?
Yes, sir, you are audible.
Yes, sir.
Yeah. Thanks, Aditi. And at the outset, I would like to mention a word of thanks to organizers, especially Dr. Sandeep Karu, for organizing this beautiful event and inviting me as a speaker. So thanks, Dr. Sandeep, for this invitation. So maybe first I would like to share my screen. Is it visible?
Yes, sir, it is visible.
Yeah, thank you. Yeah. Yeah. I would like to start my
Khandwe sir,
Yeah. Yes,
all slides are seen.
sorry.
We can see all the slides.
Okay. Now also.
You click on one number first.
Now. Hello.
Still we are seeing same.
Is it okay now?
So maybe if you reshare the screen again, then it will come clearly. Stop the sharing and then maybe you can do it again so it will come.
Sure. Is it okay now?
No, sir, we cannot see anything now.
That's good.
Yes, yes,
Okay.
yes, yes.
Yeah, I'm sorry. I'm sorry for the inconvenience. Yeah. I would like to start my presentation with, with this media news, which was floated in Indian media around 2.5 to 3 years back. So all we know, there was a huge scam in Punjab National Bank, and intensity was very high, and the scam amounted approximately to 11,400 Indian crore Indian rupees. Now you must be wondering what is the purpose of showing or giving a reference to this scam in this presentation. I just wish to highlight the fact that what missed because of such scam. Like this is exactly a cost which is required for a new drug development. We know Dr. Michel has informed us like approximately 1.8 to 2 billion US dollars are required for drug development. And probably with this fraud, entire community, the human community probably lost an opportunity of getting one more drug for the benefit of human society. So this is just I'm trying to highlight the amount of intensity or the amount required for drug development by pharmaceutical industry over here. Dr. Michel has already explained us. So what is this slide is all about? This is a typical slide which explains us the journey of drug development right from its discovery in the laboratory up to getting marketing approval by the concerned regulatory authority. So it is a time journey of 12 to 15 years with very poor success rate of 1 in 5,000 or rather 1 in 10,000 candidates, and again requiring amount as high as 1.8 to 2 US billion dollars. And that's why, since I mean, the drug development is not easy procedure. By all means, it's very, very difficult and stressful procedure. And that's why not all pharma companies dare to get into research activities. They don't dare to get into drug development activities because in true sense, they require deep pockets. And that's why there are very few companies across the globe who are into a classical drug development activities, which includes preclinical drug development and clinical drug development. Just to name few of them, starts with Johnson and Johnson, or maybe few other names include Pfizer, Roche, Novartis, Merck, Glaxo, Sanofi, AbbVie, Takeda, and company Shanghai Pharmaceuticals Holdings. So these are rich companies across the globe who are investing huge amount of time and huge amount of money for drug development. The question is, if there are few companies and if it requires so much time and so much money, what motivates these companies to get into drug development? Why? They keep themselves away from drug development. Answer is very, very simple. So this slide explains why few pharma companies who have rich or deep pockets, they to get into clinical research or drug development. This is only because of the provision made by regulatory authorities, and that provision is a patent. So what typically these companies do, these companies file a patent. So whenever they have any potential candidate, potential molecule with them, they go for patent filing. So after successful patent filing, they get a patent for next 20 years. It means the drug or the drug product or the technology with which they are going to develop or formulate their product, that will be patented for next 20 years. Of course, this 20 years include the period required for preclinical development, clinical development, and thereafter they get marketing duration. So as Dr. Michel said, in general, we require 10 to 12 years for successful development of a drug. So these 12 years are subtracted from the patented period of 20 years. but still the pharma companies get around 7 to 8 years for their marketing across the globe. And the beauty of this marketing is like during this period, there is pure monopoly. No other pharma company is allowed to sell their product or market their product during this period of patent. So what these companies do during these 7 years times, these companies sell their product across the globe at the cost of their choice. There is no regulation, there is no limitation or inhibition from the regulatory authorities. And these golden 7 or 8 years, these pharma companies make a lot of profit. So whatever amount they had invested for drug development earlier, that amount is recovered, and on top of that, they get huge profit. And that's why, I mean, this is also equally rewarding. And that's why the companies do take to get into drug development. So what is also equally rewarding, and that's why the companies do tend to get into drug development. Okay, so I said every pharma company after drug discovery, they get a patent for 20 years. So there is a monopoly, there is no other company. But what happens after 20 years? It is interesting to know, like up to 20 years, we have only one product, this innovator product or a standard product, a new product developed by a company. But once that patent period is over, once patent expires, then other pharma companies, they are allowed to formulate this drug and sell in global market. And such products are called as generic products, and such companies are called as generic companies. The earlier company, the first company which discovers drug, it is called as innovator company, and the product is called as innovator product. And the follow products, they are called as generic products, and companies are called as generic companies. So after 20 years, I mean, then we have one innovator product in the market, and along with that, we also have many more generic products available in the market. So what happens because of this? Because of this, what happens? The cost of the product comes down drastically. So when there was monopoly, when only innovator product was in market, say for example, it was getting sold at 100 rupees. Now after generic products come into market, and if there are 5, 6 generic products, the product is significantly lowered. So the product comes down by 10 times or 20 times. Then let's see what is the reason why generic companies can sell their product with such low cost. The answer is very simple, because the generic companies did not pass through all the stages of drug development. So the preclinical phase is not required, clinical phase is also not required, and only few regulatory requirements need to be fulfilled by the generic companies. And that's why they are able to sell their product at relatively lower price. So what is that remedy? That remedy is nothing but a bioequivalence study. So what regulatory authorities say, for generic product to bring for generic companies to bring their generic product in market, simply conduct bioequivalence study, and with successful bioequivalence study results, you can market your product in the respective regulatory territory. So let's see what is this all about. So same thing like here, uh, we are seeing how cost erosion occurs with marketing of more and more generic products. Say for example, here we can see when there was only one product, the cost was around 100%. With the launch of 20 products, the cost drastically comes down to 5. So there is 20 times cost erosion because of availability of generic versions in the market. So what basically the generic companies do? Their job is very, very simple. They did not discover the product. They did not spend their time in finding out the lead compound. What they need to do, The first step is simply they need to develop their formulation over their own formulation. This formulation is called as test formulation or to be tested formulation. Then next step is they need to demonstrate bioequivalence between their formulation, which is called as test formulation denoted with letter T, and they need to compare it with existing innovator product, which is denoted by letter R. And this entire, set of activities require only 2 to 3 years. Just compare, the innovator company required 10 to 12 years. Here, generic company simply requires 2 to 3 years, and that too only for two activities. First, develop their formulation, and second, conduct their bioequivalence study. And that's why cost of generic product is very, very low. So here, uh, things are summarized a bit. Like this is what innovator company needs to do for the innovator product, right from animal studies to clinical studies and then regulatory approvals. But for generic company, this animal part is not required, clinical part is not required. So this duration, this cost is saved completely. Simply they need to formulate and conduct bioequivalence study and submit to regulatory authority for their approval, and just it's a matter of 2 to 3 years. So let's see what is this remedy, what is this bioequivalence, how it works, how it helps the generic companies. So bioequivalence is nothing but it is comparison of bioavailability between the test product and reference product. We know what is bioavailability. Bioavailability is that part of drug which after administration reaches to the systemic circulation, means to the blood part. And we know it is assumed in medical science that once the drug reaches to systemic circulation, once drug reaches to the blood part, it goes to site of action and it produces desired action. It may be good action or bad action, but that is produced. So what is important in bioequivalence is to assess how much drug really reaches to the systemic circulation indirectly to the site of action. And this is nothing but it is called as bioavailability. So bioavailability in simple terms is the fraction of drug which reaches to systemic circulation, and it is always expressed in terms of percentage. So here, we can get better idea. Let's say, we are taking tablet or capsule or any other formulation by oral route of administration. So from gastrointestinal tract, it needs to cross the gastrointestinal wall. Thereafter, through portal circulation, it goes to liver. There it undergoes first pass metabolism or hepatic metabolism, and again, it goes to systemic circulation after reaching to cardiac chambers. So this is how drug reaches to systemic circulation in general. So here, same thing. Once we take tablet, it undergoes the disintegration process, dissolution process in gastrointestinal tract, and from gastrointestinal tract, it goes to liver, to heart, and to other parts of body, to systemic circulation. So here on this page, I'm trying to summarize how this bioavailability concept works. Say for example, subject or patient is taking 100 quantity of the drug. This white colored portion, let's say it's a quantity of drug. So some part during its absorption, it gets destroyed in the gastrointestinal wall itself. So let's say 30% part is destroyed or only 70% part goes to next level. Then out of that, again some part is destroyed in liver. So step by step the quantity will go on reducing, and ultimately only 10 out of 100 reaches to the systemic circulation. So when we administer 100, 10 is available to systemic circulation for its effect at the site of action. So this is nothing but a bioavailability, and here bioavailability is expressed as 10% bioavailability. Same thing, like from dosage form, drug gets in systemic circulation, it reaches to site of action, and then it produces required effects. Why I'm highlighting on so much for this particular portion? Because the entire concept of bioequivalence is based on this particular science. Like they compare test and reference formulation only by means of bioavailability. Similar bioavailability means similar effect, and similar effect means the drug is okay to get launched or approved in the market. So what typically we do in the field of bioequivalence? So like drug is given, and after drug administration, we take blood samples at regular time intervals. There is criteria of taking blood samples. I mean, which time points should be selected, for how much duration we should collect sampling points. There is criteria on the basis of pharmacokinetic properties of the drug under consideration. But typically, what we do, I mean, we give drug administration and serial blood samples are taken, their concentrations are measured, and such graph is plotted. So for Y example, we have concentration, on x-axis we have time in hours, and this is how we characterize the PK profile. So this is Cmax, maximum concentration, and this is very, very important in bioequivalence. This is one of the primary parameter for comparison between test product and reference product. Why? Because this gives us an idea whether our test formulation, the generic formulation, is not, or it is going to produce any effective level or not, and secondly whether it is going to produce any toxic level or not. So for that evaluation purpose, Cmax is one of the important PK parameters, and another important parameter is this area under curve. So we plot graph for this concentration versus time, and this AUC is calculated for test product as well as AUC is calculated for a reference product, and comparison is done between test product and reference product. And there is one more parameter, Tmax. Tmax is required for any drug to achieve this Cmax. So this parameter is not a primary parameter in direct terms, but for certain drugs where we expect a quick onset of action, there is one parameter called as AUC reference Tmax. Means the AUC observed by or demonstrated by the reference product up to this Tmax. So in this case, it will be this AUC reference Tmax. So sometimes this parameter also becomes a parameter of significance or parameter of consideration for evaluating or assessing bioequivalence. So this is simple presentation, graphical presentation for what we do in bioequivalence studies. Now, here there are two graphs. One graph is this blue color graph is for innovator product, and this red color graph is for generic product or test product. Now here what we do, we compare this Cmax with this Cmax. So this is a single point observation comparison, and we also compare this AUC versus this generic product AUC. And there are certain statistical criteria. Some difference is allowed, but if the difference is too much, usually 20% difference is allowed by and large for conventional drugs, which can be as low as 10% for narrow therapeutic index drugs, or it can be as high as 30% or even more than that for highly variable drugs not having any safety concern. Okay, I would just like to give an idea how these bioequivalence studies are typically performed uh, in pharmaceutical industry or CRO industry. This is a gold standard. This is a gold standard or standard design which is implemented across the globe for conducting bioequivalence study. What it says like you simply follow a crossover design, meaning that same participant should get test product as well as reference product. It means if there are certain factors related to the participant or related to the subject, those will be uniform or constant for test formulation as well as reference formulation. And that's why regulatory authorities are more comfortable with crossover bioequivalence study. So what we do typically in crossover bioequivalence study, basically the study population is screened. We ensure that the population meets protocol defined inclusion exclusion criteria. So after doing these eligibility assessments, subjects are divided into two groups, group 1 and group 2. Group 1, initially they get test product. After test product administration, their blood samples are taken. In subjects or these patients, they are sent back to home. And after certain duration, this duration is called as washout period. During this duration, the drug is eliminated from the body. It is ensured that washout period is adequate for complete elimination of the drug from body. So this washout period is given, and thereafter again these participants are called to the center and they are administered with reference product. So subjects who got test product in period 1, they get reference product in period 2 and vice versa. So those individuals who had reference product in period 1, now they will get test product, and in similar fashion, in period 2, we will get their PK samples or blood samples, and graph is plotted, and comparison between test and reference product is done for Cmax, AUC-T, and AUC infinity. And with statistical models, statistical approaches, 90% confidence intervals are calculated, and on the basis of acceptance criteria, the product is declared either as bioequivalent or bio non-equivalent. There is one more design or alternative design, especially followed for those drugs for which the washout period is very high. We know few drugs have very slow elimination half-life, and that's why they require very high washout period. And sometimes it is not feasible. It is not feasible for the study participants. Sometimes it is not feasible for the sponsor or CROs to retain these participants for longer duration. So the regulatory authorities have given this another option of parallel bioequivalence study design. So here what we do, the subjects are divided into two groups. One group takes test formulation, other group takes a reference formulation, and again their PK samples are taken, Cmax AUCs are calculated, and those are compared between test and reference product. And statistically, it is tested whether the test formulation is bioequivalent or not. So same thing, I have tried to present here this picture. Is a crossover design. Individuals taking in test formulation in period 1, they are crossed over to reference formulation in period 2, and vice versa. And in parallel design, individuals who are getting test product, they are never ever exposed to reference, and vice versa. So this is a gold standard crossover design. In general, I can say 95 to 97% of the bioequivalence studies are conducted with crossover design, and only 3% to 5% studies are conducted with parallel design in the field of bioequivalence. So just trying to summarize the generic players. These are not innovator companies. These are not branded companies like Johnson and Johnson or Pfizer or Glaxo or Sanofi or Abbott. These are global generic players, major or big or giant generic players, meaning that they develop their generic formulations on bigger scale and they launch their product across the globe in all the regulatory authorities territories. Just to name these giant generic players, I can say Teva, Allergan, Mylan, Cern, Aspen, and many more. But the beauty of this slide is that here in top 10 generic players, top 10 generic companies, we have 5 companies from India. Sun Pharmaceuticals, Dr. Reddy's, Lupin, Arabindo, and Cipla. So 5 companies from India, 5 generic companies from India, they are among top 10 leaders of generic players or generic companies. So this is current scenario, how this generic business is occurring at the moment. The generic business is, taking place across the globe. Not, there is not a single country who is not into generic business. Companies present in their countries, in their territories, they are into drug development also and in generic business also. Here we can see India ranks number one, followed by Brazil in terms of volume. So number of studies or uh, what I can say, the volume of business is very high in India and Brazil when we see the global scenario. And the purpose of highlighting this page or this slide is like those candidates, those students, or those individuals who are willing to join pharma industry, they can definitely give due importance this to this particular fact. They can definitely consider this generic industry or bioavailability, bioequivalence industry for their bright future. Now let's see what is Indian scenario as far as bioequivalence industry is concerned. In India, there are more than 80 clinical research organizations or contract research organizations are typically called as bioequivalence study centers. Precise number is 82, but here you can see maximum number of CROs. They are located in Maharashtra, Gujarat, Andhra Pradesh, Tamil Nadu, and other localities. They have few number of CRAs. My point is, if someone is interested to join this industry, B industry, they have very good scope to get into this career opportunity, and they can be part of these states, part of these B centers as far as bioequivalent study conduction is concerned. 20%, 20% of the bioequivalent studies conducted across the globe, conducted everywhere in the country, are conducted in India. So every fifth study is conducted in India. I'm saying this particular fact about bioequivalent study. So every fifth bioequivalent study across the globe is conducted in India. It means India has very good potential for BAB business. Number one. Number two, India can complete the BAB studies as per the standard regulatory requirements without any hesitation or problem. And studies conducted in Indian CROs in Indian setup are very much acceptable to all major health regulatory authorities such as US FDA, EMA, or Anvisa Brazil, WHO, and many more. Right. So what would be future of bioequivalent studies in India? There was some uncertainty. There were some regulatory hiccups or obstacles few years back because of lack of clarity either by the Health Authority of India. Then few facts were projected very badly or negatively by media. So that time, people were having apprehension whether this generic industry will survive or not, whether bioequivalence industry in India will be able to sustain their reputation or not. But now I can say in affirmative terms, like there is no need to have any apprehension or any worry because the regulatory authorities, they have come up with user friendly guidelines, sponsor friendly guidelines, CRO friendly guidelines without compromising with the safety of subjects, and they are giving assurance like within stipulated time frame the health authorities will give all necessary statutory approvals so that sponsors can target their timelines, they can plan their studies in India and get them completed within stipulated time frame. Similarly, in coming days, probably the Health Authority of India will ask all companies to conduct bioequivalence studies for already registered products. For a re-registration, they will make it compulsory. So it means there will be a bright future for BAB industry or bioequivalence field if someone is really interested to get into this. Right. So this is again a projection or anticipation, but here we can see China and India. These are coming up as an emerging markets for generic industry, because we know India and China both have good population, then patient population, healthy subject population representing therapeutic area is also very high. ICH-GCP trained investigators are very high. People have good exposure, good experience, and that's why in coming years, India and China today also they are leading generic markets, but in coming years also they will continue to maintain this number one, number two rank as far as generic business is concerned. So that's all about the brief orientation or basic concepts in the field of bioequivalence. Thank you. Thank you very much.
Thank you so much, Dr. Suhas, for your valuable guidance to us. So I just would like to make an announcement that our next speaker, Dr. Anand Gaikwad sir, was unable, due to unavoidable circumstances and urgency, he was unable to join today. So we would like to introduce our next guest speaker. And for that introduction, I would like to call my colleague, Riya Vayal, to introduce the speaker.
Thank you very much, Dr. Suhas Khanve, sir, for a remarkable presentation. Your preserved and wise words helped magnify our understanding in a best possible way. So moving on, ladies and gentlemen, and our distinguished guests, it is my pleasure to welcome today's next speaker, Dr. Akash Pandey, who will be enlightening us on the subject of psychopharmacology of bupropion, a novel discovery and potential implications. Dr. Akash sir is an MBBS MD in preventive medicine, passed out from BJ Government College, Pune. He has completed his PhD in pharmacology and neuroscience, his MS in medical biotechnology, both done from Texas Tech University Health Sciences Center. Sir is an accomplished patient-centric and science-driven physician neuroscientist and a people's leader. He's a pain medical affairs professional with additional responsibilities in clinical research and monitoring at clinical trials. Currently, he is the head of a worldwide team of designated physician representatives at Johnson & Johnson's, as well as personal investigator on multiple worldwide clinical studies. From Alexander D. Kenny Outstanding Graduate Awards to the late Gopal Balkrishna Titare Award, Dr. Akash is a title holder for several such honors. Along with his own distinctive published original research articles, he's an editor as well as a reviewer for various scientific journals like the Journal of Neurochemistry, Indian Journal of Critical Care Medicine, and several others. It's an honor to have you, sir, as our speaker here today. Now, on to you, sir.
Thank you very much for that elaborate introduction. Let me try and share my screen. Can you all see my screen?
Yes.
Can you see the full screen view now?
Yes.
Okay. Yeah, so really thank you very much. And it's really my honor to be here and have this opportunity to present you all. And my sincere thanks to Dr. Sandeep Kadu for inviting me And maybe many of you may not know this, but Dr. Sandeep Kadu and myself, we are classmates. We were together. We did 11th and 12th together in Ferguson College. So it's really a great honor that my old friend has invited me to present. Now, I looked at the itinerary and I saw that everyone talked about the clinical studies. And as you know, I work, and as Dr. Khandwe already identified, that Johnson & Johnson is one of the largest company in the world. And I'm really fortunate to have the opportunity to work for this company. Unfortunately, even if we are invested heavily in doing research, I will not be able to present any of our research here just because of the company confidentiality. So what I decided is that I decided to present you a really a good discovery story. That is a discovery of a new mechanism of action for already a drug that is already been marketed in the United States, and how we did that. So you all know bupropion. Bupropion is marketed in the United States under the trade name Wellbutrin. So what we'll do is that I'll just go through really high level overview of what bupropion is, what is its pharmacology, and then I'm going to give you a brief overview of cis-loop receptors. Now, what cis-loop receptors are, again, I'll talk about it later on. And then what happened, that what led us to make this new discovery. And then we'll talk about, okay, once we made the discovery, how we characterize the interaction of this drug with these receptors. So, and then at the end, we'll summarize and I'll talk about some of the potential implications. So again, this research data has already been published in the journal called Neuropharmacology, and these data are not affiliated in any form with my current company, that is Johnson & Johnson. And obviously, I may not be able to reveal some of the data because of the confidentiality. So this research was actually funded by the National Institutes of Health of the United States. And we did this research in the year 2015 when I was in academia, and I was working as a postdoctoral research associate in a lab. Now, the bupropion, as you know, it has 3 approved therapeutic indications. So first is, it is prescribed for major depressive disorder, also for seasonal affective disorder. And in the United States, it is also prescribed for smoking cessation, people who wants to quit smoking, cigarette smoking. So it is marketed under the brand name Zyban. Now, I have written here 4 mechanisms of action, but at the time, when we did this research, there were actually only 3 mechanisms of action. And why I'm saying 4, that I'll let you know. So, number 1, it's already known that bupropion, it blocks the dopamine reuptake transporters, thereby increasing the dopamine in the synapse, and then people feel good. Similarly, it also blocks the norepinephrine reuptake transporters. Most recently, I was not recently, but 20 years back, they also discovered that this drug also targets nicotinic acetylcholine receptors. As you know, this is from the basic pharmacology. Now, considering bupropion was really first invented, I would say more than 60 years back, by Dr. Nariman Mehta's lab here in the United States. And then it was marketed for depression and smoking cessation in late 1980s or early 1990s. So after that, until 1999 or 2000, it was just known to target only dopamine transporters and norepinephrine transporters. And in 1999, they discovered that it also targets nicotinic receptors. And in 2017, we published this paper and we identified its 4th mechanism of action. So now we know that bupropion also inhibits serotonin type 3 receptors. And this is the story that I want to tell you that how we discovered this new mechanism of action. Now, this research in the context of clinical studies or the design of clinical studies, which were presented to you earlier, this part of research actually belongs to preclinical phase. And there is a lot of effort. There's a lot of research that goes into preclinical phase as well, where the new drugs are discovered, new drug design and drug development process is involved in there. And these drugs are tested in vitro and in vivo, that is in animal models. And then it moves forward to the clinical phase, where it goes through the phase 1, phase 2, phase 3. And obviously, you all know there is a very high failure rate. It's a very expensive process. And that's why when the drug is marketed, particularly in the United States, it's IP protected and generally it's very expensive. So I talked about the receptors, cis-loop receptors. So you may not know that this detail, and I really don't want to get into the complexity of the molecular neuroscience, but as you know, I already told you that bupropion, it targets nicotinic receptors. So what are nicotinic receptors? You probably have learned during our second year medical school in pharmacology, how nicotinic receptors are actually involved in nicotine addiction, and at the same time, they are excitatory receptors. And but I would like to give you some more information is that they belong to this particular class, that these are membrane channels. So they are in the membrane, and when agonist hits a particular site, the channel opens and there is a current that happens in there. And that's how we have all these chemical neurotransmitters. Chemical signal is converted into an electrical signal, or electrical signal is converted into a chemical signal. That's how our brain works. So there is a highly complex neural network in our brain. So these are some of the receptors, other receptors which belong to the same category, that is cis-loop receptor. So why I'm showing you here this part is that this is where we started thinking about that this particular drug, that is bupropion, was later on found to inhibit this nicotinic receptor. There is another receptor which is similar to nicotinic receptor, that is serotonin type 3 receptor, and both they conduct positively charged ions, that is sodium and calcium. Particularly serotonin receptor, it conducts sodium and calcium, and you know those are positively charged. And if you look at the structure, this receptor has 3 parts. There is a part that is outside the cell, that is extracellular domain. There is part of this receptor that is within the membrane, that is called transmembrane domain, and the part of the receptor that is inside the cell, that is called intracellular domain. And this structure is highly conserved across this family. So different receptors from this family, they have similar structure, not identical, but similar. And that's what I'm trying to show here. You can see this part is very similar, and also the transmembrane domain is very similar. So we call it, these domains are conserved across the superfamily. So obviously, I already told you what was the compelling reason to believe bupropion would also inhibit serotonin type 3 receptor, right? What happened is that just before this research, and this research was done in collaboration with Harvard Medical School, the Department of Neurobiology, and this was actually my PhD dissertation topic. So it was my thesis. I already published it, and it's already published in the journal Biochemistry in 2012. Here, we found that we wanted to know, until 2012, it was only known that bupropion just inhibits nicotinic receptors, but we had no idea how it inhibits, where it binds, what it does, and how it actually blocks the receptor. So we wanted to identify the site where it interacts with this receptor. And there is a highly complex studies. We do biochemical methods and biophysical methods to identify, and I really don't want to get into the more details of those, but we use a radioactive compound, or radioactive or an analog of bupropion, which is tagged with a radioactive probe. And then we use that in a molecular neuroscience or basic science research to figure out where it binds. And that's when we determined that bupropion actually has two sites where it interacts with this receptor. One is in the channel and one is outside at the interface of the extracellular domain and transmembrane domain. So as I mentioned earlier, this domain, transmembrane domain, is highly conserved. It is very similar across this family. So that's when we started thinking that, hey, what about serotonin type 3 receptor? Because this part is very similar to nicotinic receptor for serotonin type 3 receptor. So both nicotinic acetylcholine receptor is cation conducting and 5-HT3 receptors are cation conducting, right? So we asked this question, whether bupropion would also inhibit 5-HT3 receptor. And this is the process of hypothesis formation. So when we talk about hypothesis into research, that's how we formulate hypothesis. So hypothesis has some rational thinking behind it, and then we think that, and then we formulate this hypothesis that bupropion could inhibit 5-HT3 receptor. Now we need to test that hypothesis, whether it is true or not. And for that, then follows an elaborate methodology and experimental process that actually is applied to identify either to reject or accept the hypothesis, right? So our hypothesis, as I mentioned again, the bupropion, it inhibits nicotinic receptors. So it very likely might as well inhibit 5-HT3 receptors. Now to test our hypothesis, we need to do some experiments, right? And figure out whether that's true. Again, I don't want to get into the complexity of this. So we all by now know what are mRNA vaccines, right? Because of the COVID. So what is mRNA vaccine? And mRNA vaccine actually for COVID actually is very discovered very close where I live in the United States. It's in Penn State University. There are two researchers, they discovered how those mRNA vaccines, and now the mRNA vaccines actually saved so many lives, right? So mRNA is actually a genetic code, right? It's a DNA which is transcribed into mRNA. It has a genetic code for a biomolecule, and when you inject that mRNA into a cell, that cell accepts that mRNA or genetic code as if it's own, and it decodes it and forms a biomolecule. Whichever biomolecule, for example, COVID, it's spike protein, right? So you have this mRNA for spike protein in the mRNA vaccine. When vaccine is injected, that mRNA is taken up by our human cells, and the human cells, they produce spike protein, and which is identified the immune system, and immune system, starts producing antibodies against this. So similarly, how do we study these receptors, right, in vitro, outside human body? So we do know the genetic sequence for serotonin type 3A receptors. So what we do is that we make mRNAs for 5-HT3 receptor. And now we don't have human cells. So what we use here is that it's a xenopus oocyte. It's a frog type of frog that we we extract oocytes, that is eggs, and these are really tiny, like 1 millimeter size eggs, and then we inject mRNA. And so this egg is a whole cell. It's a one cell system. It's not a multi-cell system. It's a one cell system. We inject mRNA, and this oocyte, it actually encodes the mRNA or the genetic sequence, and it makes those biomolecules, the corresponding biomolecule. So in this case, as you can see, after 24 to 48 hours, it makes these receptors. It makes this, produces these 5-HT3 receptors, and as these are membrane proteins, they are trafficked to the membrane and they get inserted in the membrane. And then what we do is that we apply electrodes, and this is a very tiny system. We do it under microscope. We apply electrode, and when you throw in a proper agonist, for example, for serotonin type 3 receptors, if you add serotonin, what happens is that the channels open and you can actually measure the current. So let me show you the results. So again, we wanted to see whether bupropion inhibits this channel or not, right? So we injected the mRNA, and then we started measuring the currents. And again, I don't want to get into the much details here, but as you can see here, what happens is that we use a constant amount of serotonin, right? When you add serotonin, the channels open and you can see this current. Then what we do is that we mix increasing concentrations of bupropion. So in the beginning, we use this 3 micromolar. So again, I wouldn't go into the what is micromolar and that. So 3, 10, 30. And as you can see, these are log doses, so log friendly doses that we use, so that when we plot it, it looks nice and beautiful, elegant. So as you can see, as the concentration of bupropion goes on increasing, there is decrease and decrease in current, right? So this, and when you wash it out and use no bupropion, the current reappears. And hydroxybupropion is actually a metabolite of bupropion, which also been shown to inhibit the dopamine transporter, norepinephrine transporter, as well as the nicotinic receptor. So we wanted to see whether its metabolite as well has any effect. So as you can see clearly, it also inhibited the serotonin induced currents from the serotonin type 2 receptors. And when you use this data and put this data into a statistical algorithm that we use, we mostly use softwares to do this. We don't actually do manually anything. So we plug in this data and it gives a nice dose inhibition curve. And from this, you can calculate the IC50 values, right? That is the concentration of the drug to produce 50% inhibition, right? And these are the IC50 values. So what this data shows is that we were able to actually show that bupropion, it reversibly and dose dependently inhibits 5-HT3A receptor. So once you identify that it inhibits, the next steps are really to characterize, like, is this a competitive inhibition? Is this non-competitive inhibition? Again, we are going into basic pharmacology, right? And if you know what is the difference between a competitive inhibition and non-competitive inhibition, right? Competitive is the two molecules are competing at the same site, whereas non-competitive is they are interacting at two different sites, right? So what happens is that when you have a competitive antagonist, right, you use serotonin and you get this dose response curve. But the competitive antagonist, what happens is that it starts displacing the agonist. But as you increase the concentration of agonist, the agonist completely displaces the antagonist, right? So ideally, the second curve has the same efficacy. However, it shifts to the right. That shows that it's competitive antagonist. Whereas if it's a non-competitive antagonist, obviously, if you increase the concentration of agonist, it will not be able to displace the antagonist because they are not interacting at the same site. So what happens is that your efficacy as well decreases. You cannot just expel the antagonist because the sites are two different distinct sites. And this is how we prove that this is actually a non-competitive antagonist. Now, this is a functional assay. Does it true when we check the actually binding? So we can actually test that, whether they interact with the same binding site or at a different site. And I'll just show you this one. If you look at these curves, what we did here is that, so there is a known antagonist of serotonin receptor. So we use a radioactive form of that, and then we bind it to the receptor. And what you do is that you increase the concentration of serotonin, keep on increasing, and see if it can displace from its binding. So when you use serotonin, as you can see, it completely displaces. That tells you that they are sharing the same site. And when you increase serotonin concentration, it is displacing, right? So it is just to we use it as a positive control because we know that serotonin is an agonist and granisetron is a competitive antagonist, right? However, when you use bupropion is unable to completely displace granisetron. What that tells you is that clearly they are not sharing the same site, right? So this is the another way to show that these are non-competitive, and this is a non-competitive block. So by now, I hope that I was able to really simplify this complex molecular neuroscience experimental process and be able to really convey the message or take-home message. So again, the summaries are that this is the summary of the results and conclusion. So we identified that bupropion actually inhibits serotonin type 2 receptors, and this is a I would say this is first time that we have identified. So it's a new discovery, and it has been acknowledged by many new pharmacology books, experimental pharmacology books, and soon you may see it in Goodman-Gilman as well. So we also showed that its metabolite as well inhibits 5-HT receptor, and like I said, this is an hitherto unidentified molecular target of bupropion. And what it does is that now it is going to provide a novel pharmacological basis for bupropion's effect as an antidepressant as well as a smoking cessation drug. So you would say, okay, you discovered a new mechanism. So what? Why do we care? That's the actually next question. So what happens is that in the drug design phase, once you identify a target, then there are efforts for it actually generates or gives rise to follow-up research, right? Where then we try to develop, characterize this interaction into more detail, trying to identify a 3D model of how this drug interacts with that binding site. And that actually informs further drug design, a structure-guided drug design and drug development. So, for example, bupropion is not devoid of side effects. There are many side effects bupropion can. So, I mean, suicidal ideation, or there is a problem with insomnia, right? So this actually informed us that why probably bupropion may be causing insomnia, because until now we didn't know that it interacts with 5-HT3 receptor, and as you know, 5-HT3 receptors are implicated in insomnia. So again, as I mentioned, there could be structure-guided drug design based on the identification of how bupropion interacts with this receptor, and this will lead to new improved therapies for depression and nicotine addiction. And as I just wanted to highlight it that Dr. Imad Damaj, he's considered as a stalwart in actually bupropion research. He acknowledged this, and actually these are his views, as I have quoted these, citing him. So this is not plagiarism. So in addition to its actions as catecholamine reuptake inhibitor, bupropion has also been shown to result in non-competitive antagonism of nicotinic receptor and negative allosteric modulator of serotonin type receptor. And he cited our paper, and then he states what are the implications, either of which may underlie the beneficial effects found for smoking cessation. And we believe that not only for smoking cessation, but also as an antidepressant. So this was the team when I was in the lab, and these were our collaborators. So I collaborated with the Department of Neurobiology in Harvard. We collaborated with the University of Ottawa, Pasteur Institute in France, Washington University School of Medicine, UT Southwestern Medical Center, and Case Western Reserve University. And I just wanted to acknowledge our collaborators and thank them for collaborating with us. And it's another point that I want to make is that it's not a one person that does this; it's a huge team, and that is how this humongous work of doing all these research efforts are actually done and are carried out, because there are so many people behind this. Yeah. So with this, I would like to thank you all. And if you have any questions, you're open to ask, or you can email me on this email.
Hello, thank you. Thank you, Akash, for your lucid presentation. I request Varun to deliver vote of thanks.
Am I audible?
Yeah.
A very warm good afternoon to one and all. I, Varun Gule, would like to give a vote of thanks for the day. Firstly, I would like to extend my heartwarming thanks to our Secretary General, Dr. Sadanand Sir, and respected Deputy Director, Dr. Abhijit Divte Sir, for supporting us to conduct this session. We also can't thank enough our respected Dean Sir, Dr. Sunil Natha Maske Sir, for this valuable guidance throughout. And this session wouldn't have been possible at all without the constant motivation and organization by our organizing secretary of research cell, respected Professor Dr. Sandeep Kadu sir. Lastly, we can't thank you enough, Michelle Ziong ma'am, Assistant Managing Director and Marketing Director, Gulian Pharmaceuticals, Shanghai, China. Dr. Suhas Khandwe sir, President Clinical Services, Acutus Research Laboratories, Navi Mumbai. Dr. Akash Pandre, Associate Director Medical and Clinical Services, Head of Global Consumer Medical Monitoring Organization, Johnson & Johnson, Fort Washington, USA. Lastly, thank you Dr. Raut sir and Dr. Indurkar sir for your valuable guidance and all the audience attending this. Thank you.
When Akash, I'm very personally thankful to you. Thank you for your valuable time. Suhas sir, Akash sir, Michelle madam, thank you very much. We end this meeting.
No problem. Thank you.